All right, good morning, everybody. Hope you all had a good weekend. We have a small chunk of chapter 2 left to cover, and then we will get into chapter 3. I am pretty sure we will get through all of this and have time to get into at least some introductory stuff of the next chapter. I did have one question right as we were getting started about the exam. It is open book, open notes. It will be posted on Moodle, I believe, right after lecture Friday is when it's supposed to open. We'll have more details with that. Hopefully Friday we'll have a little time for some review. With that said, stuff in the lab, quizzes, all of those things should be posted on Moodle, and they also will be open book, lab notebook, not your OpenStax book. I don't think you need that for the lab stuff. Any questions or concerns about that? Okay, so thinking lab, that's kind of a nice segue because this puts us right to one of the main ideas that we think about in microbiology labs, right? There are, and I've mentioned this the littlest bit, there are often, you will find yourselves presented with some circumstance, and the first thing that you're going to think about with that specimen is, what is it? What does it look like? You're not going to necessarily know what it is, but you want some information, right? You have a feeling that you're looking at some sort of bacteria and you want some details, right? There are all sorts of different types of staining techniques that we might employ. Sometimes something just as simple as knowing, is it a coccus or a rod shape? Might be all you really want to know, but I back it up and say, I don't know who would only want that, all right? There's a lot of other stuff we can get with some specialized staining techniques, all right? In the lab, we think about initially starting off with some simple stains where we only apply one type of a dye. You put that onto your specimen, you let it stand for a minute, usually about a minute, and then you rinse it with some water, you blot it dry, and you look at it under the microscope. And it will be like the top figure. You see the coccus or the rod-shaped organisms. Maybe they're in chains, linked together. Maybe they're clustered. You can get some information, all right? And there are numerous types of stains. We have tried to whittle down some of the stuff in the stain trays in the labs just because we're not doing a whole bunch of different types of techniques. And it's a little easier to just not stock all of the stuff if you're not going to use it, all right? But technically, like any of our gram staining reagents, we could kind of think of that, well, not any, most. We could think of as maybe just a simple stain where you could apply it and visualize something, but you're not getting an actual gram reaction. Keep that in mind. You have to go through these steps to observe a gram stain and to actually see what's happening. The simple addition of crystal violet, and that's all you get, we would call that a simple stain, right? But in a gram staining procedure, we go through a handful of steps and it gives us details that just a simple stain can't tell you, all right? There's all sorts of details about this that I think are really pretty interesting. We won't get bogged down with huge historical stuff, but a scientist, a physician, Hans Christian Graham, he was a microbiologist, but also a physician. And he was curious with organisms that were causing lung infections, pneumonia types of bacteria, and wanted ways to visualize. This is late 1800s when this staining technique was developed, all right? Few years prior, Robert Koch had come up with some techniques to visualize tuberculosis. We'll see that here in a couple of slides down the road, with a technique that we call acid-fast staining. Another series of staining procedures that you apply to the organism in order to get it to show up. And we'll talk about mycobacterium or tuberculosis in a bit, and some differences why something like what we're thinking right now, Graham stain, doesn't technically work with those guys, all right? So he comes up with this staining procedure, Koch, to visualize a different type of pulmonary infection, tuberculosis. But there are others that we weren't able to see this way. We wanted to know, what is it, all right? You could potentially see, but not get the details about it based off of some of the counter staining with tuberculosis. We'll get that in 1 second, all right? So Christian Graham comes up with these ideas that he wants to be able to visualize pulmonary bacteria, and now we use it basically to visualize all sorts of organisms, right? It doesn't matter if we're thinking bacteria, this is almost always one of the first things that occurs, all right? So the idea with this, and we will go through more details a little bit later, but when it comes to bacteria, typically, almost always, they have cell walls, right? There's one type of bacteria called mycoplasma that's an oddball and it lacks a cell wall and it doesn't stain like this, all right? But everybody else has a cell wall, all right? And then the idea is it's either really thick, numerous layers stacked on top of one another of peptidoglycan, a protein sugar combo that makes this cell wall very strong, all right? And then there are other bacteria that have a very thin, like single layer of peptidoglycan. They've got, all of them have a plasma membrane. We'll go through it here and show you the anatomy of a cell in the next chapter and give you some details, all right? All of them have a plasma membrane. Then we see cell wall material. If we're talking gram positive, super thick. If we're talking gram negative, very thin. And then another outer membrane, all right? We know this now, all right? But back in the day, we had no clue, all right? This type of a gram stain, this type of staining procedure is what we call a differential stain. It's something that allows us to tell the difference from one thing versus the other, all right? Thick versus thin cell walls. That's what we're thinking about, all right? So it's a series of steps. The lab manual will go through and describe stuff. I can tell you, all sorts of lab manuals spell this out. They are very similar. There are a few things that sometimes deviate the littlest bit. Some of it is timing, all right? I want to tell you, when you do this stuff, you need to be kind of paying attention to how long this is happening. It shouldn't just be just a quick put it on and rinse it off, all right? But it doesn't have to be exactly a minute, all right? Close to a minute, all right? Give or take a little bit, all right? What's critical is this third step, and we'll get that here in a second, all right? Oftentimes lab manuals spell out a time in seconds, and that is something you definitely should not be paying attention to, all right? You want to be watching your technique, you want to be watching your slide. And dependent upon how thick or thin of a smear you made and then stained it up, is going to kind of dictate how long it takes to decolorize those specimens, all right? And it's definitely something that takes a little bit of time, all right? You guys in intro labs aren't going to spend massive amounts of labs staining stuff up to really kind of feel like you've got a great handle on it, all right? We will give you a couple of opportunities to stain some stuff up, but it's something that we don't get super bogged down with it here. We got other things to deal with, all right? So with that said, you'll probably be focusing more on the time, all right? So after you have made your smear, you've spread it onto that glass slide, you've allowed it to air dry. I described this stuff a little bit before. You air dry it and then you heat fix it. You pass it through the flame of the Bunsen burner a couple of times to make sure that bacteria is really kind of stuck or adhered to the glass slide. After we have done that, we're going to add a series of reagents to the slide, and we're going to rinse in between each one of these steps. If you didn't heat fix the slide, the bacteria to the slide, it's highly likely that as we go through this process, you're going to rinse most all of that bacteria down the drain, and it's going to be super hard to visualize anything. All right. So the first thing we do after we've made that slide and heat fixed it is to add the crystal violet. In the lab, there'll be these little glass staining trays that go over the trough. You've all been in the lab and seen the trough in the center of the benches. Your slides will sit right on that. You apply the dye right to typically the center of your slide is where you probably made your smear. We say you kind of flood it on that, all right? It doesn't have to be spilling over the edges, but it doesn't also need to be just a single drop of stain, right? A good amount, definitely enough to cover the entire area where you made your smear, right? You put that crystal violet on and you allow it to stand for about a minute, all right? At that point, you grab the slide, you tilt it up, the stain starts to run into the trough, and there should be squirt bottles with water on the top of all of the benches by your stain trays. There's also a little knob that faucet there that will turn water on. You could use that as well. Squirt bottles work a little bit better because it's kind of a smaller stream of water. You gently squirt the water above your smear, not right on it. You're not trying to spray the smear off, right above the smear, and then the water kind of rinses down, right? In between each of these steps, we're going to rinse with water, all right? We don't show it up here, but it is a step, all right? So crystal violet, about a minute, rinse with water. And you just rinse real quickly. You see the color run off, and then it basically looks just like water running off the slide. At that point, you put the slide back onto the tray, and you add iodine. The bottles probably say grams iodine, they're probably glass brown bottles. The iodine is a special step here, and technically you could kind of use it as a simple stain as well. It's going to give you kind of a brownish color that the cells would pick up, but it's more saffron or crystal violet that we might think of as just a simple type of stain. So the iodine, it interacts with the crystal violet, all right? As the iodine and crystal violet dye interact, they form a big complex, and it becomes a massive molecule that becomes stuck inside of a gram-positive cell. It basically is retained, all right? And on the side image, we're trying to show you what would happen after each of these points if we were to visualize it under the microscope, all right? You technically don't do any of this until you get to the last step. That's really when you look at it. But what if we looked at it only after this or after the iodine? Everything still looks purple, right? That gram-negative cell is still going to retain that, all right? We haven't tried to decolorize it, all right? And the stain is still within the cell, all right? Even rinsing with water doesn't remove the crystal violet or the iodine. It's retained in that peptidoglycan layer, all right? The third step is the critical step when we're thinking about timing, all right? This should be a few seconds, all right? There are definitely things that I have read that say 20 seconds. or maybe even longer, upwards of 30 seconds. That is way too long. That's too much alcohol, all right? You will find that you can really pretty quickly make something that should look positive and purple turn into something that looks negative if you've applied way too much alcohol, right? If you don't know what you're looking at, how would you know that you've made a mistake, right? We want to make sure you know what's going on. And when we first start these things, you will be given things that you know. You know that this bacterium should be gram negative or pink, or you know that the other one should be gram positive or purple, right? Shape wise, well, we'll deal with that in a minute, right? But you kind of know what you should be looking for. And then later, being given something different that you don't know, hopefully you've gone through these techniques and you feel a little more comfortable with that, all right? But I feel like it's better off to know from the start what something is, and then when you find it, it's better, all right? So this third step, again, after the iodine, we rinse with water. Then we put the slide back down. Or no, actually, after the water, we keep the slide at an angle, and the alcohol is going to just rinse off, just like the rinse with the water, all right? The alcohol should say grams alcohol or 95% alcohol or ETOH. ETOH is just an abbreviation for ethanol or ethyl alcohol. There are sometimes another alcohol reagent that is in there for stuff like the acid fast staining, and it will say acid alcohol. You do not use acid alcohol with the gram stain, all right? It will make a positive look negative, all right? So the grams alcohol is added, the slide is at an angle, you drip or squirt that alcohol onto the slide above the smear, and you watch the color at the bottom of the slide. You will notice at the bottom of the slide that the color starts to look kind of purple, it gets a little lighter, and then it goes away. And typically a few seconds is all that's needed, all right? Once that alcohol dripping off the bottom of the slide looks pretty clear, you grab your squirt bottle, your water, squirt water above that, that rinses the alcohol off and stops the decolorizing process, all right? At that point, after the alcohol, if we looked at it under the slide, the gram positives look purple and the gram negatives, well, they look invisible pretty much. You have to be very careful when you're looking at that to even see the faint hint of a cell wall, all right? They kind of show up, but they can be overlooked. And if all you had were gram negatives and not a mixture of negative and positives, you probably might think there's absolutely nothing on there because you haven't counterstained, all right? So at this point, after the alcohol's been added, those gram negative cells with that outer membrane, that lipid layer, the alcohol touches that lipid and basically dissolves it, makes it open up. And that super thin layer of peptidoglycan, it's very easy for the crystal violet iodine complex to just get washed out. So the alcohol removes it from a gram negative, the color, but doesn't do anything to the gram positives because that cell wall is so thick. It gets in there and forms this massive complex and it can't pull it all out, right? Like I said, if you use way too much alcohol, well, it definitely can. It keeps removing and removing, and then finally you do pull all of the color out of the positive. It takes time for that to happen, maybe 20 seconds, right? So after this point, we move down to the step that allows us to visualize those negatives. And we call this a counter stain. We call the crystal violet the primary stain, the safranin a counter stain, right? The counter stain really doesn't do anything to a gram positive cell. It's a pink or reddish colored dye. Pink and purple make purple, all right? It's not going to change the color of those gram positives, right? But a gram negative that was made invisible by the alcohol, now when you add that pink dye to it, it picks it up and now you can see it, all right? So we can differentiate positives from negatives based off of their color. And again, it takes a little bit of time to get a feel for it and know that you're seeing something that's purple or you're seeing something that's pink. Seeing them side by side or in a mixture together definitely makes it a little easier. Sometimes if you use a little bit too much alcohol, these gram-positive cells are just a little bit lighter. They're fainter, right? They're a little, not really pink, but they're not super purple, right? And that makes you kind of wonder what's happened. And it really is probably just technique. It's some of your timing, right? So potentially, this is something you will find yourself doing and you will become quite proficient at making gram stains. And timing is not something you're super concerned about. So this image is a mixture, a mixed culture with gram positives and gram negatives. And the gram negatives at this point are super hard to see, all right? They are there. They are very faint. They look kind of washed out, all right, as we're looking at this. And there's a couple of things I will say that will kind of make sense as we think about this, all right? So the gram positives, a little easier to see. It also, is maybe not the best image that we could be showing, but it's pretty good, all right? So we've got a mixture of types of bacteria, Staphylococcus aureus and Escherichia coli, or our good friend E. coli, all right? Staph aureus, typical types of skin bacteria, E. coli, intestinal bacteria. Both of these two are types of bacteria that you are going to see repeatedly in the lab. We're going to use them, we're going to grow them, and we will see how they look under the microscope. There's all sorts of stuff we will do, all right? So a couple of quick things. The Staphylococcus is a coccus shape, as its name implies. They are circular. Staphylococci, oftentimes when we look at them under a gram stain, are clustered up. We showed an electron micrograph of that last time, right? And we saw the big purple staphylococci. So here on a gram stain, you can see them all kind of bunched up together. It's purple, indicating positive, right? And then all of the stuff that's kind of blurred out here is E. coli, and it is a super small gram-negative rod, all right? Sometimes when you look at it, you might think you're looking at a gram-negative coccus, but very, very little, all right? Nothing like the size of these circles here, all right? Almost pinpoint-like, all right? And there's one thing I want to tell you with that. The cocci, when they lay down on the slide, they're all a bunch of balls, right? So it's easy for them to just be there, and it doesn't matter which way that ball is on the slide, it looks round regardless, right? But if you've got a rod and you put it onto that slide, these things are super, super small, all right? You might expect, and well, probably so, the bulk of them will be laying down flat, right? So you would see these rod shapes, but some of them might be standing up on their ends. And then it looks like a little, all you're seeing is the tip of it, a little teeny pinpoint of kind of pinkish color, right? With E. coli, if it's just straight up E. coli, you oftentimes see lots of rod shapes and then little teeny pinpoint dots, right? Those dots are the E. coli standing up on its end, all right? Which kind of seems like that shouldn't be happening, but these suckers are microscopic, right? A micron in length, all right? Not so hard for them to stand up on their ends, all right? But with the cocci, it's just a ball. It's gonna look the same, all right? So keep that in mind, all right? Another thing I want to point out that kind of, well, to me, makes sense why the pink guys are so blurred out, all right? When you are focusing in on a smear, all right, when you're trying to see your stuff, you've got to pile of bacteria all over the place. And you have the potential, as you are zooming in on it and focusing in on the bacteria, you can see like the top bit, you can move through and see more on the inner, and then you can move toward the bottom. And dependent upon where you're at, as you're kind of moving through that smear of bacteria, some stuff is in focus and some stuff goes out of focus, right? The cocci is quite a bit bigger than the E. coli, all right? And we're focusing more in on these gram-positive cells. They're a little crisper, a little clearer to see, and the E. coli is out of focus, right? And if we switched it around, we could get the E. coli in focus, and then our purple stuff would be more blurred out, right? You will see that in the lab as you start seeing some of your cells, right? You can kind of see the ability to kind of move through there, right? I know in the biology lab and sometimes in the micro labs, although we don't do it, we kind of assume you've probably seen and worked with some microscopes the littlest bit. There are some slides that are already prepared that have things like threads on there, 3 different colored threads, and they're all together. And as you move through, you can see, you know, initially just looking at it, you just see something that looks kind of like a thread, but then you see that they're like together, one's on top of the other, one's somewhere else or whatever. I can't remember. That was long, long ago. But they're different, and you can see the kind of the depth of that, and that kind of puts this into perspective as well. All right. There are other details. Sometimes your smear is really thick. That makes it sometimes difficult to tell the difference between stuff. If you have a very thick smear and you're just seeing a bunch of stain, you try moving toward the edge, the fringe, the little bit where that bacteria is really faint on there, oftentimes gives you some better separation and easier to see stuff. If your smear is more thin, you try moving more toward the center where you hope there's maybe more of it kind of stuck within that, all right? And that also takes a little bit of time to just get a feel for what's happening. So this image, another one, and as we're projecting it up, it's definitely blurred out a little bit. This is just a pure culture. So instead of the mixed culture with positives and negatives, which if you're looking at something, a specimen from an individual, it's likely you're going to see more than one type of bacteria. But other times we want to see just one thing, a pure culture. And sometimes growing the bacteria in the lab, doing different plating techniques helps to ensure that what we're working with is just this one thing. And we may stain it up to verify, all right? So this is just Staph aureus by itself, a bunch of purple cocci in clusters, and it definitely is pretty easy to see how they're all kind of grouped up. This has to do with the way that the bacteria replicates. We're not going to go into details with that, at least not right now, all right? So I mentioned the tuberculosis, the acid-fast staining procedures for this were developed just a little bit before the gram stain that we showed you, right? We want to talk and introduce gram stain first because that's really what you think about doing, all right? You would not acid-fast stain every single specimen that you're ever given because it only works to stain a few things. Gram stain works to stain basically everything and then bacteria. And then the few that it doesn't, that raises some flags and you're like, wait a minute, what are we dealing with here? Let's figure it out, right? Maybe we're talking mycoplasma that doesn't have a cell wall. Some negative staining would allow us to see that. Maybe we're talking tuberculosis, and dependent upon what the patient's presenting with, if they're coming in with pneumonia-like symptoms and we gram stain the sputum specimen and we see nothing, mycoplasma and tuberculosis would both be something on our radar, right? Gram stains wouldn't pick those guys up. And then, dependent upon some of the signs and symptoms, mycoplasma pneumoniae causes walking pneumonia, all right? Very mild, if you're talking pneumonia, it's kind of a mild form of pneumonia, all right? Tuberculosis is very different, right? And the symptoms with that are going to be different. So you may be able to just kind of pick that up based off of some patient history beforehand, as far as what sorts of stuff you're looking for, right? Maybe not. And you employ both of these stains. It's only going to take you a few minutes to stain up them with either method, and then you can see what's going on, right? So the acid-fast staining, there's technically two procedures that we can use for acid-fast staining. Zeal-Nielsen, and the other one I believe is the Kinnuin method. Doesn't matter, all right? Excuse me. The deal with this, there's a couple of different reagents, and one of them, the Zeal-Nielsen, I believe, if I remember everything right, Zeal-Nielsen is the one that we would use. The steam bath, we use heat, basically to cause these cells to kind of warm up, loosen up their cell walls the littlest bit. Mycobacteria is full of a substance called mycolic acid. It's like a wax layer that makes it super hard for the stains to penetrate in. Gram stains won't, all right, because they can't go through that waxy layer, all right? But the acid-fast stain does, all right? So the hot method loosens up that wax layer and it allows the stains to penetrate in. The cold method, the Kinnuin method, uses dyes that are highly concentrated, all right? And so you don't need to use heat, but you do need to use more caution. You're doing this like in a fume hood. The dyes are something you want to kind of minimize your exposure to. In the labs, we do the hot method to back off the concentrations of all of that stuff, and you can do it on the bench top, all right? So these acid-fast bacteria, carboxylic acid is one of the, that's part of the acid staining. Carbylfuchsin, this reddish dye, the carbol is the carboxylic acid and the fuchsin is the dye. The two together penetrate into these cells through that wax layer and basically are retained. After we have stained this, and if we're doing the hot method, it's about 5 minutes of steam bath with the stain being applied to it, all right? After that, we take the slide, we rinse it with a bit of water, and then we flood it with acid alcohol. This is not something that you gently put on there like a gram stain. You squirt that stuff on there. You're supposed to be able to decolorize for upwards of a minute, all right? I have found that that seems, at least with what we've worked with, way too much still, all right? But in a real clinical specimen, you decolorize for 30 seconds to a minute, and then you counter stain with methylene blue, all right? Anything that's not our tuberculosis or our acid-fast bacteria, as we see here, is going to have a bluish pigment. So if we had more bacteria than tuberculosis in there, if there was a mixture, we would see some other stuff, all right? This is not bacteria. This is just whatever, just like stain, right? You'd see cells. So these guys we say are filamentous. This is not one bacterium. This is a plethora of bacteria all stuck together with that waxy cell wall. They want to kind of glom onto one another, all right? So you get big globs of them. Here's like an individual, it's a handful. They're kind of long, thread-like bacteria, and they typically group up. When we don't play with tuberculosis in the labs, but we do have some mycobacterium, a cousin, a non-pathogenic strain. It has the same cell wall characteristics, and when we grow it up in a flask, these cells, just like you see here where they want to kind of stick together, they form huge globs, waxy globs in that flask. And that also kind of makes it sort of an issue when you try to stain it up, because you need some of that waxy stuff, and it's hard to make it work right. All right. So acid-fast staining is it's the only time when we can take a specimen from a patient we can stain it up and we can see these acid-fast bacilli, and we can say, without a doubt, you have tuberculosis. You could do a gram stain on, whatever, and see something like this. And I know now because I read down below that it's Staph aureus and E. coli, but if you just showed me this picture, I couldn't say with 100% confidence it's Staph and E. coli, right? My gut would tell me this is some sort of Staphylococcus, right? The pink stuff, I really couldn't say, right? But purple clusters, I'm thinking Staphylococci. Aureus or epidermidis, I don't know, right? Not based off of a simple stain or gram, I shouldn't say simple stain, a gram stain, right? But this, you do know, right? Because nothing else is going to be doing this, right? There are some fungi that will pick up some of the stain. There's another type of bacteria, but we're not thinking lung infection, right? So different, right? And if we're talking this, we prescribe antibiotics immediately as soon as we see this. We still want to cultivate it. We've got to grow it up and figure out what drug is really going to be the most effective. But we start treatment immediately. The other image with the Staph and E. coli, you still need to know. You got to grow it up. You have to differentiate stuff to truly know what you're looking at, right? So pretty crazy, right? So we talked some negative staining. India ink is a classic one for some negative staining. This is something we're not going to look at in the lab, but we will talk about the littlest bit. We can use it to evaluate organisms to see things like maybe the presence of a capsule layer, a sugar coating, all right? And if these bacteria, or in this case with the Cryptococcus, this is a type of fungus, so big yeast cells up there, they have potentially, and this isn't the best looking, but you can see some where you can see the cell and then a huge halo around it. The halo would be that polysaccharide layer. That's sort of what this is all trying to show here, but it's not as good because you can't really see the cell wall of the yeast, but that's the idea, capsule layer. This is a similar idea, different type of stains, but it's trying to show us a capsule on some bacillus. So they also can produce the sugary coating on the outside, and that kind of glowing halo around them is what is really being shown here, all right? So crystal violet and copper sulfate together, not a gram stain, right? It's a different staining procedure that allows us to visualize it. Here's some more bacillus, maybe the same ones we were just looking at. This is different. These are staining up the spores, the endospores that the bacteria have. There are a couple types of bacteria that have the potential to create spores, right? For our purposes in here, we think about Bacillus and Clostridium. They are the two genera of bacteria that produce endospores. There is another one that is basically like just an environmental bacterium. It's, as far as I know, no instance of any sort of health issues. It also forms spores. I will mention it, but we don't think about it in here, all right? Sporosarcinia is the other one, all right? But again, Bacillus and Clostridium, they are the two genera of bacteria that we think about as spore formers, both of which can definitely be pathogens. The Bacillus is an organism that we call aerobic. It likes oxygen. Clostridium is a bacterium that is an anaerobe. It hates oxygen. So the details with them are definitely different. We're not going to get bogged down with that right now. All right. So these cells have the potential to form spores. All right. If you have an environment, if you have a situation where these bacteria are constantly being fed, they're bathed in nutrients, wastes are being removed, everything's all fine and good, right? you're never going to see this happening. They're just going to grow as bacillus. They're very happy, all right? But if you have them growing in a flask where it's a finite amount of nutrients, wastes will accumulate, all of this stuff is a closed system. These bacteria grow, everything's all fine and good, and then they start starving because nutrients are being depleted, wastes are accumulating, that can become toxic. There's numerous things that the bacteria can pick up, some cues from its environment that basically makes it think, if a bacterium could think, holy crap, I'm going to die here. I ought to do something about it, all right? And that bacteria forms a spore, all right? It's a protective mechanism to ensure that it can go on to grow another day, all right? They basically go through this process of sporulation where it's almost like the bacteria is going through the process of replicating, all right? It's gonna copy its DNA, but instead of copying and separating into two new cells, it's gonna copy that DNA and then it's gonna package it up. It's gonna condense it. It's gonna shove some enzymes in there that are gonna help it hatch out and be a bacterium later on. But it packages all of that up and then it coats it in a protective shell. And then once that's formed, the bacteria dies, all right? And what you're left with is just a shell of nucleic acid and enzymes that will hatch out when the conditions become favorable again, all right? So all of these suckers here with the green on the insides are in the process of forming spores. They're going through sporulation. Here's one that's already done it and the vegetative cell is completely gone. Here's a few that are still hanging out, having a good time, right? Not forming spores yet, or maybe they're thinking about it, right? Little hints, but not green, all right? So spore stains allow us to visualize the potential for a bacteria to do this, all right? Sometimes we could see bacteria, and based off of a gram stain, you might see a little hint, a little hollow bit. If it's this, and you gram stained it, these guys would look nice and purple, and there'd be hollow little circles inside, all right? And you would think, maybe I have a spore former. You do the spore stain, and then you know, all right? In this case, we've counterstained with safranin. So now, technically, they look as if they're gram negative, but we didn't do a gram stain, right? This is an endospore stain. These aren't gram negative cells, right? They appear that way with the safranin color, but they're not negative, all right? What we're looking at is the green stuff, all right? The red just shows us vegetative cells, and it's missing here, right? So a gram stain could show us purple cells with hollow bits, and maybe we see other cells that have some hollow bits, but we do a spore stain, and none of that stuff shows up, all right? That could be some sort of structure inside, food particle, potentially, that's inside of that bacterium, an inclusion body, right? Something that's brought in. It's almost thinking like a eukaryotic cell, like a vacuole, but it's not, all right? So bacteria may have some of that stuff, and a spore stain helps you to differentiate if you're truly curious about those cells. Are they spore formers or not, all right? And there are definitely differences with that as far as how they appear. Some cells swell up in the process of sporulation, and others, like our bacillus right here, don't. They basically stay the same shape. But like some Clostridium, like our tetanus bacteria, one end of it swells up real big and it kind of looks like a tennis racket. Tetani looks like a tennis racket, right? And big and green. Here's another bacillus, and this is showing some of the flagella. Flagella can be a mechanism that helps us to differentiate. We can group them up based off of how the flagella appear. Here we've got them completely covering the cell all over the place, kind of Medusa-like, I think, if as I see these. We call this paratrichous flagella. We'll go through details of this and describe all of that, but all over the place, all right? And these types of staining procedures, the flagella are very, very small. And so it's a very tedious, very fragile technique to step after step, adding stain to slowly coat the flagella to the point that you're able to visualize them. We definitely don't mess around with this because it's very difficult to get this to happen. When we see the bacillus, we just see the rod shapes. Just like these guys, there's flagella, but we're not seeing it, all right? So wrapping it up, all right, the simple stains, just one dye, right? There's basic acidic and negative stains, a handful of different potential dyes, only the addition of one, right? Could be a simple stain, all right? And giving you different details, all right? And then we move into the differentials, all right, where we add multiple reagents and rinsing in between to allow us to differentiate between stuff. Gram stains for the positives versus negatives, the acid fast to show us if the cell has mycolic acid, the spore stain, again, to see if they've got spores, flagella, or the production of capsule layers. I've got just a couple minutes. I know there's just a few slides left. This image, these are a couple of different devices, microtomes or ultramicrotomes. These are, it's kind of like a razor blade or something like that, that will shave specimens into like paper thin or thinner than that, little slices. I saw a student that was going through this process with a microtome when we were experiencing the electron microscope, and the microtome was right outside of the room and she was doing something, right? I didn't know for sure what she was doing, but it was something like this, slicing, and it was weird little white stuff. I'm like, what are you doing? Slicing bone. Was what she was doing, and every little slice that came up, and she's got this super fine paintbrush that she could take and smooth it out, and if it's a nice sheet, like a little sheet of bone, she would put that, stick it onto a slide, and if it's all blobbed up together, which lots of them do until you get through the bone to the right bit, apparently, there's lots of it that's just getting brushed away, little bone shavings until you get a nice piece. Awesome, right? Very cool stuff. And there's all sorts of specimens that in turn get prepped to be visualized like that, that are just, you know, too big in the first place to slap under the microscope. But a section of it that's sliced paper thin, that's a different story. Treponema pallidum, this is the organism that causes syphilis. Here we can see an unstained through dark field microscopy, then staining it up with bright field, and then some other over here. This is a scanning electron micrograph seeing the surface. Yes, sir.
For A, it says living Treponema pallidum. Does that have to be living seen under a dark field microscope?
I don't think it has to be living to be seen under the dark field, but with the dark field, you don't have to have your specimen like fixed and prepped and stuff like that. So you could visualize living stuff. And in this instance, although we've got it captured, you would see them if it was a living specimen, you'd see them moving. So lastly, we can also, and we showed some of the immunofluorescence before, with syphilis, we can detect with some immunofluorescence antibodies and then a secondary antibody that links up to that and it glows, right? And that, I will leave you with that. I told you I'd start chapter 3, but I lied. We will start that on Wednesday. All right, guys, have a great day.